All right. Thank you, everyone, for the opportunity to present some of the work we've been doing with these mid-density genotyping platforms. And so you've already heard from Dongyan and Manoj today. And so while you're going to hear some similar language and some of the similar processes that have been used in other species, I'm going to then talk about how we've then applied this to a drought tolerance breeding program. Now, often referred to as the queen of forages, alfalfa serves as a foundational element in American agriculture. It's a versatile crop that is an ideal food source for dairy cattle, horses, and other livestock. Its deep root system not only helps improve soil health, but also protects water quality, making it a sustainable choice in many regions. Alfalfa's economic footprint across the U.S. is truly impressive. It ranks as the third most valuable field crop, generating an estimated $8.7 billion each year. Its cultivation spans over 23 million acres across the country, providing a vital source of income for farmers and supporting rural communities. Now, traditionally with alfalfa breeding, it's really relied on making decisions by eye. And so you can use genetic resources to try to expedite some of these selection decisions. But in alfalfa's case, accessible genetic resources had been lacking up until this point. Now, traditional SNP markers have several limitations when used with alfalfa. First, the marker density can be low, meaning that not necessarily within specific regions do you have enough loci, but in general coverage throughout the genome. Now, you can use tools like whole genome sequencing, but that leads into the second point that genotyping costs can be high, which can make it prohibitively expensive to genotype large numbers of plants. Finally, traditional SNP markers that provide low density may not be able to detect important traits, because if you don't have markers in a certain region of the genome, you're not going to be able to successfully identify those genome trait associations. Now, this is where we led to the development of the DART tag panel, because it's a targeted, reproducible, low-cost option for sequencing in alfalfa. Particularly because a difficulty with traditional SNP markers is because alfalfa is an autotetraploid. Now, Breeding Insight, in collaboration with USDA ARS alfalfa breeders and Diversity of Raised Technology, have developed a 3,000 SNP DART tag panel that overcomes the limitations of these traditional SNP markers. The DART tag panel was developed using a whole genome skim sequencing approach, and this approach involves sequencing the genomes of 40 elite alfalfa lines used in North America breeding programs, and then identifying SNPs that are common in the population. The DART tag panel is cost-effective and targeted, and it can genotype large numbers of plants and is able to detect important traits. Now, these 3,000 DART tag loci were selected primarily because they were distributed fairly evenly throughout the genome and their location in genic regions and for being polymorphic. Now, unlike genotyping by sequencing, targeted amplicon-based genotyping technologies such as DART tag have low missing data rates and query the same exact loci in all samples across genotyping projects, allowing new data to be easily appended to existing data. And this was highlighted earlier by Dongyan when she discussed the microhaplotype database that's being developed for species within the USDA ARS, like alfalfa here. Now, the DART tag panel was then validated using sequencings of different F1 and backcross populations of alfalfa. The validation showed that the panel is able to accurately genotype different alfalfa populations. And this panel can then be used for a variety of applications, including marker-assisted selection, linkage map construction, genome-wide association studies, and genomic prediction. In the bottom right corner is a figure from a recently published manuscript outlining this work, and this is showing the successful construction of a linkage map using those 3,000 DART tag loci. Now, again, this information that I quickly summarized, the development of this 3,000 DART tag panel was published in Genetic Resources this past fall or past summer. And so my collaborators here are listed. And if you have any additional questions or want some additional information about how this DART tag panel was developed, please refer to that manuscript listed here. Now, we then wanted to apply that 3,000 DART tag panel to a drought tolerance population. So this drought tolerance population is a half-sib population developed by nested associated mapping, resulting in 436 alfalfa half-sib families that were selected for sequencing. Now, sequencing was done in bulk So there were 6 individuals per family for sequencing. So in order to develop that drought tolerance population, it was there was a first the F1 individuals were developed from breeding a drought resistant line with a drought susceptible parent. Then those F1s were back crossed to the drought susceptible and drought resistant parents, resulting in the final nested associated mapping population that we sequenced for DART tag. Now, a thing to note about the DART tag sequencing is we discussed these 3,000 DART tag loci. Now, with these targeted 3,000 DART tag loci, we're not only obtaining information from those specific loci, but within that 81 base pair region surrounding that targeted region. So an example shown here is a target SNP is identified from DART tag sequencing, but then we're also getting information from SNPs within that 81 base pair region on either side. So in addition to the 3,000 SNPs, you can also obtain up to thousands more SNP information from the DART tag panel. So for this population, we sequenced 436 samples. And then using DART tag sequencing, we obtained approximately 6,000 loci. Now, I then took the read count information from those 6,000 loci and performed dosage calling using UpDog, and then following quality filtering resulted in about 3,000 loci. Now, the figure on the right is showing the distribution of these approximately 3,000 loci throughout the alfalfa genome. And so while we see that there's fairly consistent coverage throughout, there is some gaps listed in chromosome 6. Now, this isn't necessarily unique to DART tag because when we use other sequencing methods like GBS, this region within chromosome 6 also results in some gaps. Now, the phenotype data from this was the total harvest yields for 3 growing seasons from 2020 through 2022. The raw values were then converted to blue values in R using SPATS and AS-REML. And then these 3 growing seasons covered 5 months. This was May through September. Now, to perform the GWAS analyses, we used GWAS poly, and then as input, we had those blue values, and then the dosage calls from DART. And what we found after testing for 6 models and using a Bonferroni corrected p-value were 21 significant QTLs related to yield traits using those DART tag SNPs. Now, here shown is the Manhattan plot for those 3 total yields. And we find that there's significant loci across the genome across those 3 total yields. This black line here is the significance threshold. So any SNPs located above that black line were considered outlier loci. Now, I want to highlight here in chromosome 8, we're finding this same region considered significant across the 3 different yield traits. Now, while we don't go in depth into it here about what this may mean or what the genes are related to these specific loci, we will be producing comparisons between GBS sequencing of the same population and GBS or in DART sequencing of this population, and then we'll go into further detail about these outlier loci. Now, we then performed genomic prediction using that DART tag SNP dataset. This time we used GROWN, package in R for the genomic selection runs. Genomic selection was performed for each trait, and then 10 cross validations, 10 reps, and a 90-10 train test split was used. Now, the Pearson correlations were a calculation between the observed phenotype, this were those blue yield values, and genomic estimated breeding values. We found overall, when comparing RR-BLUP and G-BLUP models, that they were staying fairly consistent with a range from 0.139 Pearson correlation to 0.330. And again, while I don't go in depth into these analyses here, we are going to then go into further detail when comparing GBS to this DART tag data. So in conclusion, the 3,000 SNP DART tag panel is a valuable resource for alfalfa breeders, and it has the potential to revolutionize alfalfa breeding by making it more efficient and cost-effective. Importantly, this DART tag data, or this DART tag sequencing method, is a targeted approach and it's repeatable across sequencing runs. And as shown here, it can be successfully applied to existing breeding populations. Now, I'm just really the spokesperson for a lot of this work here. In collaboration with USDA ARS Alfalfa and Forage Genetics team, a lot of scientists at Breeding Insight, did a lot of the work, especially the development of that dart tag panel, and then the development of that breeding population. But I also want to highlight the tools for polyploid methods that we used in these analyses that really wouldn't be possible without that work. If you have any other additional questions or details you want to know about some of the ongoing work at Breeding Insight, also please visit breedinginsight.org, where you'll be able to see some of the other species projects that are ongoing. And so with that, thank you for your time.
Thank you very much, Alexander. Any questions? Well, a few minutes. Okay, Jeff.
It looked like you showed on your comparison of the RR bluff and the G bluff.
You want to come up here and talk? I'm not going to be able to repeat that one. No, that I won't repeat. You're on your own.
I was wondering, I was surprised to see it looked like you showed differences in accuracy under cross-validation with G-BLUP versus the RR-BLUP, even though those are mathematically equivalent methods. So is there something else going on here that explains why you don't see the same result?
Yeah, that's a great question, right? You'd expect these to be identical. Now, I'm not sure, haven't gone into this in detail, why we might be seeing these differences. If I may speculate, it could be due to the 90-10 train test split and cross validations maybe between these different models, because I don't think we're necessarily testing the same samples with RR-BLUP and G-BLUP when they're doing the train test split, since it's random between these subsets. So that could be why we're seeing differences. Now, when we compare, I'm not showing here, but when we compare across the full 16 harvest months, this difference rate here is kind of an outlier in this total yield. We see them basically lining up when we have all 16 harvests across the 16 months.
Yeah, that's when you're comparing methods, it's good to basically block them and use the same folds when you're doing a methods comparison like that to eliminate that source of variation. So thank you.
Yeah, thank you.